So as you can probably tell from my thick Philadelphian accent, I'm not from Philly, but I've been working at Penn for the last couple of years. I'm a hematologist and a hempath pathologist by training, and I've spent the last few years working in the lab of Sarah Gill. So we're a translational lab with a focus on hematologic malignancies with a special interest in AML. And I'll be talking about today how we're sort of thinking about using this new technology to better improve outcomes and diagnostics for our patients in the AML sphere. So for any non-clinicians in the room, just very simple terms, a patient with leukemia will be diagnosed, they'll be risk stratified, they'll have some sort of treatment, and then they'll have some sort of response. And what happens downstream from that depends a little bit on what happens with the disease. They might have additional consolidative therapy, they may go on to have transplant, but a lot of the time we're relying on the presence or absence of minimal residual disease to help guide us in terms of these decisions. And obviously if the patient has a macroscopic relapse, it's very clear what's going on, but what we're more interested in is being able to better understand what's going on at the more micro level to better understand what is the best treatment for our patients. Many of you will be familiar with this kind of table, and the purpose of showing it is not to go through everything in detail, but just to point out to things that many of you in the room will be familiar with, the limitations of the sort of current gold standards which we have, specifically multi-parameter flow cytometry, RT-PCR and next generation sequencing all have their advantages, but none of them really are able to give a full picture of exactly what is going on at a very sort of granular level. Traditionally, flow cytometry will give you information about an immune phenotype of the cell, which might gear you towards a particular immune-based, or in my case, cell therapy-based therapy, whereas cytogenetics or other genetic molecular type information will gear you perhaps towards a more targeted therapy. And what we're interested in sort of understanding from this DNA and protein sequencing technology is, can we actually combine these, and can we get more accurate information about what is actually the underlying driver of the cell, how that correlates with phenotype, and how we can best treat and also monitor these patients. The other reason that this is really important, and I'm not going to talk too much about this today, but just to sort of put this as an idea in people's head, leukemia has come a long way in the last decade, and there are actually now a lot more treatments available. In addition to transplant, novel chemotherapy, new small molecule inhibitor type things seem to be popping up every day. Monoclonals, BiTEs, things like that, and in the case of our lab, cell therapies and immune-based therapies. So getting it right and understanding what actually is driving the disease is sort of more important, I think, than ever. So before I get into what we've actually done, I just wanted to put it in the context of what we're trying to emulate. So we've done a pilot study, and what we're trying to do is combine elements of these figures, which will be familiar with most people in the room. So on the left is a multi-parameter flow cytometry plot, which is sort of the current gold standard for a lot of MRD testing. And the picture on the right is from a very famous paper talking about clonal evolution and clonal shift in AML. And so just keep these figures in your mind when I describe the experiment we did. And this is basically what we were trying to reproduce as a way of starting to validate how well this technology would perform. So what we did was we took 5 different AML cell lines, which are listed here. These are pretty standard ones that are used a lot in research. And we selected them based on their known mutational profile. And we did this by looking through the literature and choosing cells that had unique mutations, and the green tick means we were able to find them in 2 separate decent publications, because there is some variability when you read about cell lines' mutational status. We then looked to internally produce some flow cytometry data, and this is a pretty simple stain. So what you're looking at, the cell lines you can see running sort of on the left axis with the antigen of interest indicated at the top, and that's just gated against forward scatter. And from that, we were able to create sort of an expected protein expression for all these cell lines. To simulate leukemia, what we did was to basically try to reproduce a clonal population of blasts with mixtures of these cells of known genotype and phenotype. So for the first experiment, we wanted to keep things reasonably simple, and we just combined 2 of these cell lines, which are the HEL9217 and the OCI AML3 cell line. They were combined at a 1-to-1 ratio, went through the Tapestri sequencing pipeline, And then the output from this is how do they cluster in terms of protein, how do they cluster in terms of single nucleotide variants, and how do they cluster in terms of copy number. And then as a second part, we did the same thing, but with a more complex mixture of cells, again, keeping that sort of 1-to-1 ratio, so we knew what we'd put in, and we could try to correlate with what had come out. This is some data just about the experimental design from the Mission Bio side. So at the top is the 20-gene AML panel, which is what we used. And the protein custom panel was selected to sort of have an AML focus, though there are other antibodies which are available to use. And on the right is a summary of the metrics. So it was analysed on a high-seq, and sample 1 is the 2-cell mixture, sample 2 is the 5-cell mixture, and reasonable performance in terms of number of cells that we were able to successfully analyse, and the read depth is all shown here. So I'm going to start with the 2-cell data because it is a little complicated to look at, and I've tried to sort of use the same colours, so hopefully that projects okay. So as expected on the left, you can see that these cells separate very nicely by SNV, which makes sense because they were selected based on their genotypes. We also can see reasonable separation of CNV. The protein clustering is not as clear as we'd expected, although there is some overlap between the immune phenotype of these cells. The other sort of output that the Mission Bio team were able to help us put together from their bioinformatics team is a really nice summary slide, but it's a lot of information at once, so I'm just going to walk you through it in parts. So the top panel is the SNV data, and you can see the colour scale on the right, with dark blue being wild type, light blue being a heterozygote, and that sort of aqua colour being a homozygotes for the mutation. And you can see each horizontal line is a read from an individual cell, and you can see that the cells cluster really nicely in terms of their origin, so whether it was the HEL or the OCMI cell. And at the bottom is the specific single nucleotide variation with the location of the gene that this mutation was identified in. The CNV data is shown here, and again the scale you can see at the right, where yellow colour means gain of copy number, dark blue is loss, and that sort of bluish colour in the middle is neutral. We're working on ways to get the resolution of this better, but again you can see some clear copy number loss and gain trends emerging from the cells. And then finally is the protein data, and I've got another slide to show this in a little more detail, but again you can see there are more ubiquitously expressed antigens which don't give you perhaps as much information, but if you can imagine this in a real situation where the percentages of the populations are different, I think this data would look a little more compelling. Looking at flow cytometry first, because that's something as a pathologist was sort of my first interest of seeing how it compared. We took the side-by-side flow cytometry plots, which are the same ones I showed you on the earlier slide, and then compared how they performed with the protein expression data. Now, a caveat to this data is that we're not comparing apples with apples. We're looking at flow cytometry, which is designed to separate cells into positive and negative, whereas the protein expression data is going to be less binary in that way. So we're sort of working with Mission Bio as better ways to express this in a sort of slightly artificial way. If you put a line through and you line the plots up right, you can see that there is, again, reasonable correlation. The ones that stood out as being perhaps not quite as good with the CD123 and CD135, it may be an issue to do with the clones that were used were not the same. The ones we used for staining as the ones we used for sequencing for sort of technical reasons. But with the exception of these two and within the limitations I've already outlined, certainly reasonable correlation between the two. This is the protein mapping in more detail. So the clustering figure on the left shows you the cell which both of these populations refer to. And again, that yellow colour maybe is not particularly easy to see, but if you take, I think the most interesting example is obviously the CD33 is the standout as being the sort of the most strongly expressed on the HEL92, but basically negative on the other. And the CD117, if I can just flip back a slide, if you look at CD117 in the HEL cell line, you can see there's this sort of dim partial expression across the populations, which is the same thing that we see on the 117 on the HEL plot on the right. So again, we see pretty good correlation with these two technologies. So the second experiment follows the exact same design. The data is obviously a little more complicated to look at. So this is the multi-omics plot. And again, I will go through each one in detail because it's a lot to take in. But again, we see the same pattern that, as expected, the SNV clustering is certainly the most defined. You can see these cells, which were chosen by genotype, cluster really nicely. The C and V I'm going to go into a little more detail with that one population that sits out really nicely. And then I'll go through the protein data as well. And just for your reference, the sort of colour code on the top right, the colours chosen there match the SNV clustering just because they were the easiest to follow. So this is the SNV data, and again, it's sort of nothing that you would particularly expect. You can see the cells cluster and separate really nicely. We're in the process at the moment of going through and validating that these mutations found reflect what is in the published literature and potentially doing that sequencing work ourselves. I don't have that to show you today because it's a work in progress, but definitely a very nice separation of the genotypes. The CNV data was quite interesting, and the cell I wanted to focus on is this HEL92 1.7 cell line. You can see it sits completely separately, so this is the sort of mustard-coloured blob on the right. Focusing in on the gain of copy on that cell line on the top row, these were just two. Again, we're in the process of sort of validating that these copy number variants reflect what is published in the literature. But RUNX1 and U2AF1 are certainly both described in the literature as having gain of function. Again, this I think needs a little bit more work to be more useful in really nailing down a particular cell population. But certainly in this pilot work has been concordant with what we were expecting that we would see. And again, looking back at flow cytometry, and I apologise again, this is a busy slide with the same caveats as I mentioned previously. We're not comparing apples with apples, but looking across the board, you can see that there is definitely a reasonable correlation with the protein expression that we have seen. Our plan for the next iteration of this is to see if we can actually get the exact same antibodies for the same clones, specifically if they're compatible with the Mission Bio platform, and see if we can expand the panel a bit more to get better MRD type flow data to match the MRD data that could be generated by this platform. Again, the ones that kind of stood out as being not quite as good were the same ones I mentioned either. And we were having email discussions last night with one of the team from Mission Bio about ways to potentially improve this for the next run with both of these antigens, so CD135 being FLT3 and CD123 being a fairly ubiquitous AML marker. These are ones we're certainly interested in pursuing. And lastly, this is the protein cell mapping by cell antigen. You can see that some cells cluster more nicely than others, and I think that's got to do with how unique or otherwise that particular cell population may be. If you think about multi-parameter flow cytometry, this is going to be prone to the same potential issues, as in a patient may not have a lap, they may not have a distinct population. But if you think about being able to map a particular population of cells by phenotype and also match that with genotype, I think gives it more power than we have currently with the tools available. So in conclusion, the work we've done is a pilot study which was designed using AML cell lines of known surface phenotype and known mutational genotype with the intention of trying to simulate a multi-clonal AML population and then correlate the data we had from published literature and our own surface phenotyping with what the output of the Tapestri was. And this protein and DNA sequencing enables a resolution and correlation of individual cell or subclone, which is what we're trying to emulate, with individual cell phenotype and genotype. And again, trying to imagine that in what it would look like in a real patient is in the context of a clonally heterogeneous leukemia. Our future directions, which I think you'll hear about some from our next speaker, is looking at our primary cell analysis. Our focus in this sort of first iteration, as I mentioned, is in the MRD framework and how we can really understand what status a patient's leukemia is in. But looking further down the field, it certainly has potential application in terms of precision medicine and having more tools available to treat patients with relapsed leukemia and deciding, based on this DNA and protein set of data, what is the best and optimal way, not only to monitor them, but to treat them. So I wanted to thank my boss. So Sargil is the smiling guy with the folded arms there, and also Olga, who's one of our senior scientists who did the bulk of the wet lab side of the work. The sequencing core at Penn were fantastic. We had some dramas at the 11th hour getting this data through, but they were wonderful. The Mission Bio team, especially Robert and Sean, who've been helping us sort of from the earlier days, and in the last few weeks, the bioinformatics team have been really wonderful in helping us put this together. And finally, just to acknowledge our funding sources. So I'll finish up there. Thank you for your attention. I'll be happy to try to answer any questions you may have.